welcome to my channel. My name is Lao Sun. In this channel, I will talk about some stories that happens in the biotech industry and some of the current issues that are related to our daily life. It is a chill and semi-professional channel. My main goal is to get my point across and we can both learn something. If you think my channel is interesting, please leave a thumb up and subscribe my channel. In the US, we have 3 vaccines distributed right now, which are Moderna, Pfizer, and Johnson & Johnson. Pfizer are mRNA vaccine, and Johnson & Johnson is adeno-associated virus AAV-based vaccine. We'll talk about mRNA vaccine first, which is Moderna and Pfizer. One of the key parts for mRNA to be working is that we have its lipid nanoparticles, also known as LNP, which contains 4 major components: ionized cationic lipid, cholesterol, helper lipid, and stabilizer particles. Cationic lipid are water-loving, and inside the cationic lipid are mRNA. The difference between the 2 vaccines are not dramatic, and they have the similar efficacy as well. As you guys may notice, there are 2 areas of LNPs are different, which are ionized cationic lipid and stabilizer particles. Together with the mRNA, they made these 2 vaccines distinguishable. mRNA technology and LNP delivery have been studied and developed for more than a decade. This is one of the reasons that COVID vaccine can be developed so quickly. Before we jump into the mechanism, let's talk about how this virus is infecting your body. If you get infected by SARS-CoV-2 virus, the surface protein from the virus will interact with ACE receptors on the lung epithelial cell. Then the virus will incubate itself in your body and eventually break out and cause pneumonia. This is how you find out you get Covid-19, which stands for Coronavirus Disease 2019. mRNA works similarly in terms of mimic the spike protein production utilizing our own cell machinery. The mRNA is coated with spike protein and encapsulated in the LNP. Then the mRNA containing LNP enters the cell and forms a structure called endosome. mRNA escapes from endosome and delivered to the ribosome and turn them into spike proteins. These proteins are considered as antigens so that our immune system can attack. One important note for this mechanism is that the mRNA doesn't go into the nucleus, and there's no scientific evidence to prove that the mRNA will interact with the host DNA. This is why it is safe to use. In addition, the dosing for these vaccines are also different. Pfizer is 30 microgram in 0.3 milliliter, and Moderna is 100 microgram in 0.5 milliliter. This is why you cannot interchange your vaccine in between doses. Then the spike protein is secreted from the ribosome, and some of them reach to the surface of the cell. Then your B cell saw it and started to recognize it. It sees the spike protein as a foreign subject, and it starts to get paranoid. Like, what if I see them again? What should I do? Blah blah blah. Then it starts to produce lots of antibody in your body to prepare for the future attack. So then, when the real virus stop by, they can be recognized by the antibody and neutralize the virus. Another route after the B cell recognized the surface protein, it started to talk to the helper T cell. Then helper T cell will talk to antigen presenting cell. The spike protein will be engulfed in the antigen presenting cell and present on the surface. I will then activate the cytotoxic T cell. So that in the future, when the real virus that infected the cell, it contains the same antigen, it can be recognized by the cytotoxic T cell and destroy the virus. So now let's talk about J&J vaccine. J&J vaccine using AAV as a delivery tool for the vaccine. AAV is a very small single-stranded DNA, and the vaccine spike protein DNA is recombined on the AAV. It won't replicate itself as it is a replication incompetent AAV, and currently there's no evidence to prove that it can cause disease that integrate to the host genome. The key component of this vaccine is the AAV vector contains spike protein DNA, and the supporting components are just like the one we see previously for the mRNA vaccine that contains acid, salt, and sugar. The mechanism of AAV delivery is very similar to LNP delivery. After the AAV gets into the cell forming the endosome, instead of going to the ribosome, it goes to the nucleus. The DNA forms a structure called aposome in the nucleus, and the aposome won't integrate it into host cell DNA, and the sequence was also coded as replication incompetent so that it won't replicate itself. Then the DNA will transcript mRNA that codes the spike protein and send the mRNA to the ribosome. mRNA will be translated in the ribosome to express spike protein. Some of the spike protein was expressed on the surface of the cell and would be recognized by the B cell and follow the mechanism that was previously mentioned. After people understand how mechanisms work, then we can move our study to discovery phase and clinical trial. Generally speaking, there are 5 steps before FDA approval: discovery and development, preclinical research, clinical research, which is clinical trial, FDA drug review, FDA post-market drug safety monitoring. However, you may get disappointed after you hear this. Currently, all vaccines are not FDA approved yet. They are being used under EUA. EUA stands for Emergency Use Authorization. So EUA authorities allows FDA to help strengthen the nation's public health protections against chemicals, biologicals, radiological, and nuclear threats, including infectious disease, by facilitating the availability and use medical countermeasures needed during public health emergency. In other words, the FDA needs more time to evaluate the whole submission of the drug product, but people need the vaccine, so FDA can authorize the unapproved medical product for emergency use. The validation and process are still the same. The difference is now people can use vaccine after the clinical trial phase 3. Then you may ask a question: is this vaccine safe to use? The answer is yes. Let me give you a comparison. Pfizer, Moderna, and J&J are not yet approved, and their study groups more than 30,000. Polio, measles, and Hep B vaccines, they are approved, but the study groups that they were using for testing were somewhat different. As we can see, polio vaccines has been considered as the largest clinical trial in the history, that it has more than 600,000 people in the study. Measles used about a similar amount of people in the clinical trials as we can see in the COVID vaccine. Whereas hepatitis B, in this case, we're talking about the brand Indirax B. They used only about 5,000 people, but in 36 different clinical trials. All the clinical studies designed are randomized, double-blinded, placebo-controlled studies. So my point is, the larger the clinical trial is, the more information we'll be able to get, but sometimes it's not necessary. As long as we get the important information across and validate it, such as efficacy and safety and etc. In addition, we need to trust the FDA and CDC officials because they are the group of experts that working in this field for more than a decade. Just think about it. A group of MD and PhD scientists who study for this subject for more than 10 years, and then they present the data to the public and a scrutinized review by these two regulatory organizations. Why would you not trust them? Then people may ask, which vaccine is better? In order to answer that, we need to understand one thing first, which is efficacy. Using Pfizer and J&J as an example, there are a total of 43,000 people in a Pfizer study, 21,000 with treatment group and 21,000 with placebo group. There were 8 people in the treatment group and 162 in the placebo group got COVID 7 days after the second dose. So if you use 8 divided by 162, you got 4.9%. So this number means that there is a 4.9% of chances for people who receive the vaccine that can get COVID-19. In other words, there are almost 95% of the chances for people who receive the vaccine that won't get the disease. Well, people may ask, does that mean that 100 people got vaccine, then 5 people will get sick? No, this number is applied to each individual, which means that if you receive the vaccine, you will be 95 less likely to get the disease compared to the people who didn't. J&J did similar clinical trials in 3 different regions: US, Latin America, South Africa. It achieves overall 65% efficacy against mild to severe COVID-19 infections. Then you may ask, is J&J worse than Pfizer and Moderna? The answer is no, and you cannot compare the vaccine just by its efficacy. The bottom of the blue rectangle is a scale that represents the goal of vaccine. Well, the ideal situation is our vaccine can cover the whole range of the rectangle, which means once you had a vaccine, you wouldn't have to worry about it. But the reality is, if you have moderate to no symptoms, it's more like a cold feeling, which means your adaptive immune system will be covered that. As a result, for this pandemic, the goal is actually to cover the range from the death to severe symptoms. And if you have moderate symptoms after you get the vaccine, you probably will be able to recover it on your own. In addition, the efficacy and safety clinical trials were done by those 3 companies in different time frame and locations. The Pfizer and Moderna was done in August to November 2020 in the US, whereas J&J did the study in 3 different regions in the world between October 2020 and 2021. So this is why Pfizer and Moderna has more efficacy rate, because they did the study in the summer when the COVID-19 was harder to spread compared to the winter. J&J, on the other hand, can catch more variants because it did in multiple locations and they did the study in winter. So using efficacy alone to judge which one is better is not accurate. Instead, we need to focus more on the later part, which is the number of severe symptoms, hospitalization, and death after you receive the vaccine. And based on the clinical trials, there were 0 cases for all authorized vaccine. In April 23rd, FDA paused the recommendations for J&J due to the blood clotting disorder, and after a few weeks in early May, it resumed its administration again. There were 6 people out of 7 million who have administered for J&J shot got a blood clotting event. So this is why FDA paused the vaccine and conduct investigation. So CDC reviewed all the available data and giving the following conclusion. Reports of adverse events following the use of J&J vaccine suggest an increased risk of a rare adverse event called thrombosis with TTS. Nearly all reports of this serious condition, which involves blood clot with low platelets, have been in adult women younger than 50 years old. Based on the risk and benefit, CDC suggests that J&J vaccine has potential benefit outweigh the non-potential risk. That's why it got reused again. Human body is weird, and that's why people are still trying to figure this out. Just like what we have mentioned previously, 6 out of 7 million have blood clot. It is extremely rare cases, but some people may get it, and sometimes it's inevitable. This is why FDA regulates the side effect for all submission drugs. This happens because we have not understood human body entirely yet. This is why we need more people like myself who is willing to dedicate a career into biomedical science. So the purpose for me to make this video is also enforce myself to learn something new. This one is just an overview for Covid-19 vaccine, and the following videos will be cover the topics on clinical trials in much greater details. So okay, thank you very much for watching, and I will see you then.